Welcome back, dear scientist to be. Today, we're diving into the essential follow-up to our PCR episode, Gel Electrophoresis. It's how we actually see the amazing molecules we're working with. Get ready to sort DNA like a pro. And we're back. A quick heads up. Did you know over 80% of you watching right now aren't subscribed? If you love what we do, hit that subscribe button and join our genetics community. It really helps. Now let's get back to the bench with Cellie and Karyo. Safety is paramount. Gel electrophoresis involves electricity and potentially hazardous chemicals, so always consult your lab's specific safety guidelines and MSDS for all reagents. Always wear your personal protective equipment. Lab coat, safety goggles, and gloves. Remember, electricity means dry hands and never touching the gel or buffer when powered on. UV light requires UV shields or goggles for eye and skin protection. For stains like ethidium bromide, ETBr, it's a known mutagen and potential carcinogen. Handle with double gloves in designated areas and dispose of all contaminated waste properly. Prioritize safety to protect yourself and your samples. Gel electrophoresis is actually super clever. Imagine we have a mix of tiny invisible molecules, like DNA pieces, and we want to separate them. Our trick? We make them run a marathon through a microscopic obstacle course. That's our gel. The basic idea is simple. We place our molecules into this gel, then apply an electric field. Because our molecules carry an electric charge, this field pulls them through the gel. The smaller molecules are like tiny, speedy runners who zip through the obstacles much faster and farther than the larger, lumbering molecules. It's all about size and charge working together for separation. So what happens? Our mix of molecules gets sorted. The result is that they spread out across the gel, creating distinct lines or bands that we can then visualize. Each band represents a group of molecules that are all about the same size, having traveled the same distance. All right, dear scientist to be, let's actually run a gel. It's a fundamental lab skill, and here's how we assemble our molecular sorting machine and put it to work. First, we get our gel prepared. For DNA and RNA, our go-to is agarose gel. Think of it like a jelly-like sieve with microscopic holes. We melt our agarose powder in buffer, then carefully pour it into a mold. We insert a little plastic comb to create those wells where our samples will sit. As the gel cools, it solidifies into that porous network, ready to act as our molecular sieve, physically hindering larger molecules. We can even change the size of these holes by changing the concentration of the agarose. For example, a 0.8% agarose gel has bigger holes for separating large DNA molecules, over 1 kilo base pair, while a 2% gel has much smaller holes, perfect for separating tiny DNA fragments under 500 base pair. For a typical mini gel, like 50 milliliters, you'd mix the appropriate amount of agarose powder into your buffer and microwave it in short bursts until fully dissolved. Next, chamber setup. Once our gel is solid, we carefully remove the comb and place the gel into the electrophoresis chamber. This chamber has an anode, that's the positive electrode, and a cathode, the negative electrode. Then we fill the chamber with our running buffer, like TAE or TBE. This isn't just water. Its key roles are to conduct the electricity evenly across the gel and to maintain a stable pH. Why pH? Because extreme pH could mess with our molecules, especially proteins. Then comes sample loading. This requires a steady hand. We carefully pipette our samples, mixed with that handy loading dye, into the individual wells. Remember that loading dye isn't just for color. It makes our samples nice and dense, so they sink perfectly into those little wells. Plus, it gives us a visible dye front that runs ahead of our molecules, tracking their migration and telling us when to stop the gel before our precious DNA runs right off. Typically, you'd add about 1 to 2 microliters of loading dye per 5 to 10 microliter of DNA sample. And for comparing sizes, we use a DNA ladder or molecular weight marker. This is a mix of DNA fragments with already known sizes. It's our trusty size ruler to estimate the exact size of our unknown sample bands. Now it is time to run the gel. We connect the chamber to a power supply, making sure the DNA will run towards the positive electrode, and hit start. You'll typically set the voltage between 80 to 120 volts for a mini gel. You'll see bubbles forming at the electrodes, and the loading die fronts start to move. That means our molecules are migrating through the gel matrix. Their speed, remember, is dictated by their size, their specific shape, and their charge. Remember, DNA has a negative charge, so it naturally zooms away from the negative cathode and towards the positive anode. A typical run can take anywhere from 30 to 60 minutes, depending on your voltage and the size of DNA you're separating. And finally, visualization. Once the die front has migrated far enough, like 75% down the gel, we turn off the power. We remove the gel and soak it in our DNA binding stain. For DNA, the classic is ethidium bromide, which glows brightly under UV light, typically at a concentration of 0.5 to 1 micrograms per milliliters for 15 to 30 minutes, followed by a rinse or destain in water. But there are safer fluorescent dyes now, like S-Cyber green or gel red, that can detect incredibly tiny amounts of DNA, as little as 20 picograms. For proteins, we use different stains like Kumasi blue or silver stain. Once stained, we place the gel on a UV transilluminator, and bam! Clear, interpretable bands appear, each one representing molecules of a specific size. At the molecular level, the dye has intercalated with the DNA, allowing the bands to fluoresce under UV light. So, you've got your beautifully separated bands on the gel. The analysis phase is absolutely crucial for interpreting the results and extracting meaningful biological information. It's all about visualizing, identifying, quantifying, and sometimes even further processing those separated molecules. Now you might be thinking, why do we need this after PCR? That's a fantastic question. After performing PCR, that amazing process of amplifying specific DNA fragments, gel electrophoresis is commonly used to analyze those amplified DNA fragments. This allows researchers to confirm the presence, size, and even the quantity of their PCR products, and it's also brilliant for detecting genetic variations or mutations. The band analysis, the pattern and intensity of those glowing bands, tells us everything. It indicates the presence or absence of our amplified DNA, confirms its size by comparing to our ladder, and even gives us the relative quantity of PCR products. The brighter the band, the more DNA we probably have, allowing us to estimate DNA concentration with reasonable precision. For mutation detection, specialized gels and conditions, like conformation-sensitive gels or denaturing gradient gels, can actually reveal tiny single-base differences or mutations by subtly altering how the DNA fragments migrate. It's like finding a single wrong letter in a giant book just by how fast it moves. Next, identification and characterization. The position of each band is precisely compared to our molecular weight or size standards, our trusty ladders that we ran alongside the samples. This lets us confidently estimate the size or charge of our separated molecules. And for complex samples like proteins, specialized gels and techniques like two-dimensional 2D electrophoresis or tandem polymerized gels can even resolve different protein isoforms, like reveal post-translational modifications, like phosphorylation, providing incredibly detailed characterization of samples. For quantification, we can measure the intensity of each band using densitometers or imaging software. This allows us to actually estimate the amount of DNA, RNA, or protein in each band. And for super precise quantification, especially of DNA modifications or protein post-translational modifications, scientists even use advanced techniques like in-gel digestion, followed by mass spectrometry. And the journey doesn't have to end at the gel. For further processing and applications, specific bands can be carefully excised or cut out from the gel for even more detailed analysis. This might include sequencing that specific DNA fragment, sending proteins for mass spectrometry, or performing enzymatic digestions right on the extracted band. In proteomics, those complex 2D gel patterns can even be compared to massive protein databases for identifying and characterizing proteins in complex mixtures. Truly amazing. Even in the best labs, things don't always go perfectly the first time. Gel electrophoresis has a few common pitfalls. Knowing how to troubleshoot them will save you time and samples. Here are some frequent issues and their likely causes, along with quick fixes. No bands visible. This is often the most frustrating. This usually means there's no DNA in your sample, perhaps due to degradation, or a failed PCR amplification if you're analyzing PCR products. You might have incorrectly loaded the sample, missed the well, or there could be a power supply malfunction, no current applied. Also, check for incorrect staining or visualization. Maybe the wrong dye was used, the UV transilluminator is faulty, or you simply forgot to add the stain or used an incorrect concentration. To fix this, rerun your PCR if needed, re-verify sample presence and integrity, reload carefully, check all power supply connections and settings, and confirm your stain is active and correctly used, ensuring the UV light is working. Faint bands. Your DNA is there, but barely visible. This typically indicates low DNA concentration in your sample, underloading, you simply loaded too little sample, insufficient staining time, or even over-destaining if you're using ETBR gels. A diluted ladder can also make your sample bands seem faint by comparison. To fix this, try loading more sample if possible, increase your staining time, or reduce destaining time. Always ensure your DNA ladder concentration is correct for comparison. Smiley or frowning bands. The bands are curved instead of straight. This is often caused by uneven temperature across the gel due to excessively high voltage, which makes the middle of the gel run faster, causing a smile. It can also be due to uneven buffer levels in the chamber or an incorrectly poured gel. To fix this, reduce your voltage for a longer, more even run. Always ensure your buffer is level in the chamber and pour gels on a perfectly level surface. Blurry or streaky bands. Instead of crisp lines, you see smears. This usually points to overloading wells with too much DNA, which overwhelms the gel's resolution. It can also be due to DNA degradation, too many nonspecific PCR products, protein contamination in DNA samples, or gel issues like bubbles or uneven polymerization. To fix this, dilute your samples before loading, ensure your DNA integrity is good, optimize your PCR conditions to reduce nonspecific products, clean your samples if protein contamination is suspected, and always pour gels carefully to avoid imperfections. Distorted ladder or samples. Bands appear skewed or irregular. Common culprits include salt in the wells, which can affect migration, air bubbles trapped under the gel, uneven gel polymerization, bent electrodes in the chamber, or simply your samples running off the gel because the run went too long. To fix this, ensure you're using proper buffer concentration, carefully remove any bubbles when setting up, check your electrode alignment, and remember to stop the run before your precious samples migrate completely off the gel. Remember, troubleshooting is part of the scientific process. Don't get discouraged if your first few gels aren't perfect. With practice and attention to detail, you'll become a gel electrophoresis pro. Wow! From simply sorting molecules by size and charge to confirming PCR products and even detecting tiny mutations, gel electrophoresis is an indispensable foundational technique in molecular biology and biotechnology. It literally allows us to see the invisible world of DNA, RNA, and proteins. The journey into the amazing world of genetics is just getting started, and you're right here with us, dear scientist-to-be. So, if you're as fascinated by the incredible world of biotech as we are, hit that subscribe button right now. And don't forget to ring the notification bell so you never miss a single revolutionary discovery from the Geneticist. This episode is just one part of our exciting How to Biotech Lab playlist, so dive deeper there for more incredible insights. And for all the detailed research and citations we mentioned today, you'll find them conveniently in our Telegram channel. See you in the next episode, inside the amazing world of the cell.